me in the back?
Yes.
All right. So thank you so much for coming today. It is the last day of our inaugural Rebel Grad Slam week of competition. As you know, the Rebel Grad Slam is a 3MT competition. And a 3MT is basically a 3-minute thesis competition. So let me tell you a little bit about the 3MT competition, where it started, and why we're doing it. So 3MT started at the University of Queensland in Australia in 2008. It was, by all accounts, a ravishing success. And so they spread, the 3MT competition spread around Australia and New Zealand in 2009 and 2010. By 2011, it had made the leap across the sea, and it had spread to Europe and to the United States, and it's been growing ever since. So now the 3MT competition exists in more than 17 countries, and there's more than 175 competitions like this one that go on all around the world every year. The neat thing is that lots of schools like us do the 3MT competition on site and then partake in regional competitions with other universities in the area. And so this is obviously our first time out the gate, but by all accounts it's been a big success thus far. So I think it's safe to say that we'll return next year and we'll explore the possibilities of maybe challenging Arizona or California or maybe all of them to do a regional 3MT in the future. So why is UNLV doing a 3MT now? Well, it's a pretty cool event, and I've known about it for quite some time, and I really felt like it would be something that would be well received here and would bring a lot of benefit to the university. Fortunately, the grad college team, our leadership on the graduate council, they all agreed and supported this, as did the GPSA. And so together, we sort of put this idea into motion, and here we are. Basically, the 3MT is especially helpful, I think, to graduate students because it really emphasizes a skill that graduate students really need to have, but that very frequently don't get trained in, and that is the brief presentation of their research in everyday terms, right? It's one of those things where everybody's family is confused about what it is you study and what is this thing called graduate school, right? You find yourself on an elevator chit-chatting with somebody and they say, oh, what do you study? And there's that moment of paralysis, right? And so whether it's in everyday situations like family or friends or elevator chats, or whether you're trying to convey the value and significance of your research to a non-specialist audience who might be considering you for grants or for a job or for other kinds of professional opportunities, this is a skill that's worth having, right? And so we thought this would be a way to help our graduate students develop this skill and do it in a way that's really fun and that brings students and faculty from all over campus together, which is something that we don't do enough, right? And so in the spring semester, you know that we have the very formal research forum. That is run by the GPSA and the Graduate College, and we have about 150 to 200 graduate students each year doing very traditional presentations, which is really important. Posters, panels, etc. And that's a long-standing tradition here. And so the 3MT is really a way to take students and their research and highlight graduate education in the fall semester. So we'll have one event each semester that really brings graduate research to the fore. So that's why we're doing the 3MT. The 3MT is also part of a larger campus initiative. Many of you know that we are rated a Carnegie High institution. That puts us in the top 4.5% of universities in the nation. Not too shabby, but we have higher aspirations. We aspire as a university to grow into a Carnegie Research Very High institution in the next 20 years. And that's a heavy lift, and it's going to happen because of the hard work of our faculty, because we grow our graduate, especially our doctoral programs, because we increase the amount of research and productivity on campus. And lots of folks and lots of offices across campus are working on ways to do that. So the Graduate College, likewise, is looking for ways to become a Tier 1 graduate college and to support the university's mission. And this is one piece of that model. So what have we been doing this week? We started with preliminary rounds on Monday, Tuesday, and Wednesday of this week. We had 3 rounds each day, and we had 67 graduate students competing in the preliminary rounds. The top 3 winners of each of those rounds progress on to the semifinals. So we had 27 folks compete yesterday in 3 rounds in the semifinals. The top 3 winners of those 3 rounds yesterday were invited back today, and they're sitting with us in the front row as our 9 slammers of the day. So they have made it. Students. We've never done this on campus, so they're sort of the first ones to do this quite publicly at UNLV, and they've obviously done it extraordinarily well. So The 3MT really takes a graduate student researcher and puts them in front of an audience, a friendly one like you, and gives them exactly 3 minutes. You'll see we have a timer that runs to the left of me here, and 3 slides, which are prepared in a PowerPoint behind me. And so each of these students has 3 slides and 3 minutes. No props, no notes, no nothing. If they go beyond the 3 minutes, they are disqualified from competition. Okay? And in that time, they need to present to you what it is they study, why they study it, how they study it, and really, why does it matter? Why should we care? The so what statement. Right? And so these 9 folks will be doing that in just a moment here. Now, the reason that we got here is we had more than 40 judges volunteer all week. And without them, this wouldn't have been possible. And so on your handout, you also have a list of all the judges to show our appreciation. But those 40 judges were campus leadership, graduate faculty, administrative faculty on campus, and community supporters. And for that, I'm very, very grateful. They're the folks that helped us get here to this final event. Let me start by introducing the 9 competitors, the 9 slammers for today. We have Jessica DiBerardinis from engineering. Want to stand up and give a wave? We have Jessica Nave-Blodgett from liberal arts. Chris Hardy from the College of Sciences. Caitlin DiBenedetto from the College of Liberal Arts. Survi Sharma from the College of Sciences. Erica Marty from the College of Engineering. Daniel Mast from the College of Sciences. Sharon Young from the College of Liberal Arts. And Kate Porter from the College of Sciences. And let me introduce our judges. They really have the hardest task of the day in some ways. Wait till they see these 9. They're incredible. So the judges today are Executive Vice President and Provost John Valerie White. Dean Brent Hathaway from the Lee Business School. Monica Lounsbury, Vice Provost for Research and Faculty Development. Research, Faculty and Policy. Apologies. Elizabeth White Nelson from the Department of History. She's also the chair of the Graduate Council this year. Donovan Conley from Communication Studies, and he serves on the Graduate College Executive Committee. Virginia Selwood, who is a community supporter of UNLV and the Graduate College. And Alfonso Ayala, from administrative faculty from undergraduate admissions and recruitment. Thank you all very much for being here today. So, without further ado, you've heard enough from me. I will make one other plug, and that is we are going live on social media, Facebook and Twitter. If you'd like to live tweet or promote or celebrate and cheer on your friends who are competing, we're hashtag Rebel Grad Slam, of course. So now, Associate Dean Kendall Hartley will moderate the slam. Thank you.
Okay, as he said, I'm Jessica, and I will be doing my presentation on predicting diabetic ulcers through dynamic measurements. Now, the American Diabetes Association reported that in 2012, 29.1 million Americans were diagnosed with diabetes. This is roughly about 1 in every 10 Americans. Of this patient population, about 15% will develop foot ulcers. Foot ulcers are painful sores that occur on the bottom of a diabetic's foot that, if they are left untreated, or if they become infected, can result in amputation. It's been estimated about 1 in every 30 diabetics will need amputation due to ulcers. Now, obviously, foot ulcers are a huge problem that could potentially be not only painful, but life-changing, and thus there's a need to be able to predict and help prevent these ulcers from ever occurring. Recent research has shown that tissue stiffness increases in diabetic patients. Now, what this means is that as a diabetic walks, the deformation that they would see in their foot tissue would require an abnormally large amount of pressure. Now, this increased tissue stiffness leads to increased risk for ulcer development, and thus I propose that if I can measure and locate the tissue increase, I can then use that to predict ulcer development. In order to do this, I need a dynamic experiment. Now, what I mean by this is that it's kind of impractical to measure a person while they're standing still, because this doesn't represent their lifestyle. Instead, I want to measure the patient as they're moving, thus being able to measure the stiffness that will be better representative of what they would experience in everyday activities. To do this, I've created a setup that uses 2 cameras, seen in orange, a force platform, outlined in red, and a lighting system. Now this setup will image and measure the forces and the diabetic or non-diabetic patient as they walk across the platform. This setup, as the diabetic and non-diabetic patient walks across the platform, will measure information that I can use to calculate the deformation and the force values required to make that deformation in the foot tissue. This information will be fed into a computer algorithm, which will then calculate the stiffness of that tissue in the diabetic or non-diabetic foot. I will compare the 2 groups to determine if there is a difference between them, and if there is, what that difference is. And I will call this difference the stiffness threshold. This stiffness threshold will help me be able to predict when ulcers occur in diabetic feet. And I theorize that I can use this and the location of an increased stiffness to be able to predict ulcers in diabetic patients. And if I am successful, I can then tell the physician when and where the ulcer will occur. Now, if I am successful, I will be able to help prevent ulcers in these diabetic patients and thus help maintain the normal lifestyle that these patients so desperately deserve and prevent all the pain and suffering that these patients could endure. Thank you for listening.
When we hear a piece of music we like, we often start to clap or dance along with it. How do we do this? Even without any formal musical training, humans are remarkably good at finding the beat in a piece of music and synchronizing with it. This ability to extract a steady pulse from a complex piece of music is a form of rhythm perception. Rhythm is the patterning of events in time and is a crucial part of auditory communication systems, like speech and music. When we think of rhythm, we usually think of rhythms in sound, but there are visual rhythms all around us too, in the hand and face movements of a speaker, or in the body movements of a dancer or a musician. Strangely, visual rhythm perception has not received as much empirical attention as auditory rhythm perception, and what little we do know suggests that we're better at hearing rhythms than we are at seeing them. A recent study found a link between rhythm synchronization ability and reading readiness skills in preschoolers, but we're not really sure why. A growing body of evidence suggests that there's a link between formal musical training and improved verbal skills in children and adults. Rhythm perception may be the underlying mechanism that links these auditory communication systems together. I researched the development and characteristics of rhythm perception in audition and vision. I do this through studying musical meter perception in children and adults. Musical meter is the hierarchical organization of events in time into repeating patterns of strong and weak events. I use 2 levels of meter in my research: the beat, what we tap or we clap to, and measure, the grouping of these beats into a repeating strong-weak pattern. No one's previously examined if we're able to perceive multiple levels of musical time all at once, or in different senses. In my auditory research, I have people listen to a click track and a piece of music together and tell me how well they feel they match. I found that adults with and without formal musical training are able to do this task, but they do it in slightly different ways. Musicians treat beat and measure level information equally, whereas non-musicians rely mostly on the beat and only pay attention to the measure if the beat matches between the click track and the music. Now I'm moving into visual rhythm perception, and I've created a new visual click track. It looks kind of like a clock. I'm asking people to tell me how well my clock track matches the music they hear. If they're able to get the measure and the beat information out of the clock and match it with the music, I'll have found some of the first empirical evidence of cross-modal meter perception. There's still so much for us to learn about rhythm and temporal perception in audition and vision. A better understanding of the underlying mechanisms could help us improve and come up with treatment systems for people with problems with language and music. Understanding the different way we perceive auditory and visual rhythms could answer a fundamental question about how we actually process time in our brains. While there's still so much more to learn about rhythm perception, we do know one thing for certain. Humans have definitely got the beat. Thank you.
Hi, My name is Chris Hardy, and I'm very interested in studying obesity because it's one of the biggest healthcare issues that we face today, and it's a growing problem. In just the last 30 years, the rates of obesity around the world have nearly doubled. And that's a big problem because obesity is known to lead to things like diabetes and heart disease, which ultimately lead to a shorter lifespan. And the tendency of westernized societies to eat more and exercise less is a huge problem when treating these diseases. But science has shown that there's more to obesity than just what you eat and how often you go to the gym. There's clearly a heritable genetic component. And understanding what genes make you fat and how that fat affects the heart is the main focus of my research. In order to answer these questions, I use a very important model organism, the fruit fly, Drosophila melanogaster. And when I tell people that I study fat flies, they usually think I'm crazy, but flies are much more similar to humans than people might expect, with about 75% of genes that cause a disease in humans also being found in the fly. You can even take fruit fly insulin and activate the mammalian insulin receptor and vice versa. So here at UNLV, we've established a very unique system in order to study obesity in fruit flies. It started 15 years ago when my advisor, Dr. Alan Gibbs, took a set of fruit flies from the wild and brought them into the lab for a long-term evolution experiment. Each generation, he would select for only the flies that could survive the longest without food. And these tended to be the ones that would store the most fat. So over time, it became survival of the fattest. Each generation, fat genes were passed down. So now, some 88 generations later, we've evolved fruit flies that are extremely obese, so fat that they even have a hard time trying to fly. And so when I came into the lab, my first project was to understand if they also suffered from heart disease. And they do. We took advantage of a machine that optometrists use to look at heart structures, or look at nerve structures in the eye, and used it to look at heart structures in the fly. So looking at the results from this, we can see in a skinny normal fly, a heart that's open, completely contracts and opens over time. In our fat flies, the heart is wider, it's dilated, and it doesn't completely contract. It's less efficient, and this is a condition known as dilated cardiomyopathy. We went on to show that this is caused by extremely large deposits of fat tissue in our fat lines that physically push the heart away from where it's supposed to run along the dorsal cuticle up here. We zoomed in on this fat tissue into individual cells, and we found that these cells contain extremely large lipid droplets. Lipid droplets are the part of the fat cell that stores all of the triglycerides, which is the main storage product when you eat a lot of calories. And what's really interesting for us is that lipid droplets are very well conserved genetically in humans. Humans also have lipid droplets. It's found in our adipose tissue, and this is one of the main reasons we get fat is because these lipid droplets really swell up. So if we can figure out what genes are causing these lipid droplets to become so big, we can then have therapeutic targets in humans. And we started doing that. We started a genome-wide association study where we isolated DNA from both a skinny and a fat fly, and we sequenced it. We now know all the A's, T's, G's, and C's that make up a skinny versus fat fly. We've created a list of candidate obesity genes, and those are going to be targets for therapeutic intervention in the future. Thank you.
In the spring of 2014, Nevada was front and center in almost every major U.S. media outlet due to the standoff between Cliven Bundy, a cattle rancher, and the Federal Bureau of Land Management. Stripping away the nuances, the core of this argument was over access to land, particularly whether Bundy had the right to use the land for his cattle herds. This is only one example of many where access to land serves as a catalyst for conflict. Why does this matter? Well, it matters because this is not a new issue. Rather, conflict over access to land has plagued humans for the last 12,000 years, starting with the so-called Neolithic Revolution. This momentous development first began in the Near East and refers to the transition from hunting and gathering, which characterized 99% of human existence, to the domestication of plants and animals. And this slide illustrates some of the earliest animal domesticates in the Near East. It's really for the first time in human history that access or control over land became absolutely critical, because without it, these early farmers and herders could not maintain their livelihood. I'm interested in the movement of people and animals across the landscape, or animal husbandry or herding practices, and what these can reveal about human-animal relationships. My dissertation research will use primary data from the early Neolithic sites of Ias Yorkis on the eastern Mediterranean island of Cyprus. The site function is currently unknown. We do have these circular cobbled structures, which indicate a degree of permanent habitation to the site, but we do not yet know why people and animals were there, how they moved around the surrounding landscape, or the relationship with other Neolithic people on the island at this time. To address animal husbandry practices, My research uses carbon and nitrogen stable isotopes from bone collagen and the carbonate component of molar enamel from both domesticated and wild animal species. The basic premise of this technique is that the food consumed by the animal is used in the construction of body tissues. Bone collagen reveals what animals ate closer to their time of death, and the molar enamel reveals what animals really consumed at the earliest part of their life history. So to reconstruct animal husbandry practices, I will look at the variation between these isotopes, both between the different animal species as well as within the same species. And one of the things that this can reveal is whether or not the animal was raised locally or if it was from another part of the island and then brought to the site. This research is important because it sheds light on human-animal relationships and animal husbandry practices, both of which we know almost nothing about for the early Neolithic on Cyprus. Furthermore, it provides hope. In a time where we're literally 24/7 bombarded by news that talks about terrible things like ISIS and the Ebola outbreak, it demonstrates that for at least the last 12,000 years, we have survived and seemingly thrived, facing very similar problems. Thank you.
So the motivation of studying biology is to understand the cause of and to identify the cure of all diseases. A disease can be defined as a state in which a system fails to function properly. The human body has different levels of organization within itself, and at the base of these levels lies the fundamental unit of life called a cell. Individual cells work together for the good of an entire organism. And this working together is called as the communication between the cells, which is achieved primarily through a special class of molecules called as proteins. Studies have shown over and over again that it is the failure of proteins to function properly that leads to a diseased state at any given level of organization. Scientists have understood this concept well, and they have utilized this information in drug designing. Currently, more than 50% of the drugs target proteins. Yet, we don't have cure for all the diseases. And this suggests that we do not understand the molecular functions of proteins completely. And the reason is that there are about 47,000 proteins in a cell, and each protein interacts with 20 more proteins for a variety of different reasons. And then those 20 proteins will interact with 20 more further downstream, thus creating a giant network of interaction within a cell. The specificity of interaction within this network is achieved primarily through special regions which are present in the proteins called as motifs, highlighted in color in this image. The preliminary studies done in our lab have shown then when these motifs are present in a specific region of a protein called as the C-terminus tail, it imparts functionality to that protein. So if you chop off this region from a protein, the protein will not perform its function. And each protein has only one C-terminal tail. Based on these observations, we asked to what extent the C-terminal region is exploited functionally by proteins. And our hypothesis is it is to a very large extent. To test our hypothesis, we generated a pool of novel C-terminal motifs for all the human proteins in a cell. And then to test our predictions from that pool of 100 novel motifs, we picked a few that we are currently testing experimentally in laboratory settings. One of the motifs that we are working with, we hypothesize that it has a role in brain-cell communication. All this information, the existing information on the C-terminal motifs has been consolidated in our web application, the C-terminome. And the new information that will be generated through experimentation will also be updated in the C-terminome web application. Thank you.
Think about the number of times each day you turn on a faucet. You expect to get clean water. You don't stop to think about how it got there or the work that went into getting it there. The truth is, we take water for granted, and we don't have an infinite supply. For the last 3 years, 30% of our country has been under drought condition. And the two states with the most acreage and exceptional drought are California and Nevada. In addition, we're facing increased population. So how do we continue to provide enough water that's dependable and drinkable? My name is Erica, and my research involves water reuse. Take a look at these 3 glasses. If you were thirsty, which would you choose? Now, I don't need to be a mind reader to know you're not going to pick number 2, because when it comes to water, less is not more. So we're left with 1 and 3. They look the same, right? The difference is glass 1 comes from a typical drinking water plant, and glass 3 is from a water reuse plant. Reuse, like reduce, reuse, recycle. Sounds good, until you picture a toilet. Now, to be fair, the water in glass 1 is also reused. Unless you live on a mountaintop, you live downstream from another city's wastewater treatment plant. But that's not the end of the world. We have excellent wastewater treatment technology. We can remove particles on the level of a micrometer, like bacteria, and on the level of a nanometer, like viruses. So you certainly don't have to worry about getting things in your water like toilet paper. But water reuse does pose some challenges. And when we use a disinfectant like ozone, we know that it will kill pathogens via chemical oxidation. But that same disinfectant reacts with other molecules in the water. And these unwanted side reactions form harmful disinfection byproducts, or DPBs. Now, treated wastewater has more substances in it, so we're forming new DPBs, like the carcinogen NDMA. In the last 15 years, we've learned a lot about which disinfection byproducts will form. But what we still need to understand is the chemical process for how they form. And that's the area of my research. If we can understand this black box, then we'll be able to prevent the formation in the first place rather than removing the material afterwards, which takes both time and money. Now, so far, I've opened the door of this black box, and I've been able to identify key chemical structures that lead to the formation of this carcinogen. So at the very least, we know which substances, we can predict which substances will form it. So why should this matter to you? Where we're headed is potable water reuse. In this past year, there have been 2 facilities in the US in Texas that opened for direct potable water reuse, and more are already under construction. So while you may not be thrilled about having to drink the water from glass number 3, you can rest assured that environmental engineers like myself are committed to bringing you water that's dependable and drinkable for the future. Thank you.
Thank you. So what are extreme conditions? Well, they're anything that aren't what we experience here at the surface of the Earth under regular days. So an atmosphere of pressure from above us and the heat from below and above. So what I'm looking at is the behavior of radio elements, actinides and technetium, under conditions that are found in the center of the Earth. So thousands of degrees hotter and thousands of millions of atmospheres higher in pressure. So to generate these conditions, we use these things called diamond anvil cells. So basically, I take 2 diamonds and put them in an opposing geometry, and I put a very, very small sample in between and squeeze it. So we use diamonds to squeeze things to get the pressure, and then we use laser systems in this simultaneous configuration to come in through the diamonds to create the temperatures. So diamonds for pressure, lasers for temperature, and then we need to observe what's going on under these extreme conditions. So to do this, we take the diamond anvil cell with the sample to a synchrotron or a neutron source where we can look at the atomic structure or magnetic properties as they change in real time under these conditions. The material that I'm putting under these conditions is technetium, which the really important thing for you guys to know is it's radioactive and you don't want it in your drinking water. I don't want it in my dinner. It's just not good to eat, for the whole radioactive thing. So, if it's radioactive, it should decay away and it shouldn't be a problem, right? Except that we keep producing it. We have nuclear reactors in this country. There's nuclear reactors around the world. And every year we produce kilograms of this material. And what do? We put it in a barrel and put it out on the front lawn in front of the nuclear reactor. Does that sound like a good place to put radioactive material that likes to get into our water supply? I don't think so. But we don't have a good answer for what to do with it because it is very mobile and it does like to transport through the aquifers. So what I'm doing is using diamond anvil cell technology and these extreme conditions to look for new technetium compounds. So during my first year, I've worked with this material here on the right, which is technetium dioxide. When I squeezed it and heated it, what I saw is that these straight chains here, well, their response to pressure was to form a zigzag arrangement to become more compact. And I don't know if this is going to be the world-changing compound that prevents technetium creeping into our water, but it's something different. It's something new, and it's something we haven't tried yet. So in an effort to maintain our nuclear storage facilities and keep technetium and other fission products out of water, food, etc., we are looking at these compounds and other compounds in a novel way that can't be achieved here in the laboratory. Thank you.
As a biomedical anthropologist, I study health and disease in evolutionary and cross-cultural perspective with an emphasis on reproduction. So my research focuses on the emerging practice of human maternal placentophagy, which is the ingestion of the placenta postpartum by the mother. Now this might sound a little strange, but humans are actually the rare exception among mammals, since mothers of most mammalian species actually ingest the placenta enthusiastically following birth, but most human moms, as you know, don't do this. So we don't know when or why humans stopped, but this hasn't been documented as a traditional practice in any human culture. But in the late 1960s and early 1970s, you start to see accounts of this behavior among women in industrialized countries emerging in the literature, and it seems to be part of the natural birth movement. Now, this practice seems to have grown in the last decade or so. You may have even heard of women like Kim Kardashian or Holly Madison going on record saying that they ate their placenta after giving birth. And women now are typically steaming, dehydrating, and encapsulating the organ to be taken as a postpartum supplement in the weeks following birth. Now you might be asking why they might do this. So as these charts show, they think that there are actually benefits to this. Advocates of placentophagy suggest that taking placenta capsules during the postpartum period can restore hormones and nutrients that are lost during childbirth, and that it provides a host of benefits to the recovering mom. The primary reason that they do this is that they believe it will prevent or relieve postpartum depressive symptoms, but they also think that it can do things like improve breast milk production, help you bond with your baby, decrease postpartum bleeding, and the list goes on and on. So the question I always get is, does this really work? And the answer I always give is, we don't really know. So my dissertation research aims to address this question through a double-blind, placebo-controlled study that compares the experience of women taking their dehydrated and encapsulated placenta to the experience of women taking a dehydrated beef placebo. So as you can see, I meet with participants 4 times across pregnancy and postpartum, and at these meetings, I collect biological samples for hormonal and nutritional analysis, and I also collect psychological data that can be evaluated across the study period and then compared between groups to identify any differences. Now, I'm wrapping up the study right now, so I don't have any conclusive results at this time, but there are a few cases that seem kind of interesting and might suggest that there could be effects on mood for some women who take placenta supplements. So once this research is complete, it will contribute to our understanding of postpartum health and recovery in general, but it will also give some insight into the effects of taking placenta capsules as a postpartum supplement for recovering mothers. Thank you.
Hi, my name is Kate Porter, and I study bacterial evolution. Now, every time I say that to someone, it's usually followed by a comment like, hasn't evolution already been proven by Darwin? They say this to me like they're breaking bad news, as if the concept of Darwin's evolution has never been explained to me before. But nobody panic, that is not the case. Darwin's evolution follows the idea that mutations arise randomly during growth. If these mutations are beneficial, they get passed on to the next generation. Thus, each new generation is stronger than the one that came before it. However, my project focuses on that flat line at the top of our bacterial growth cycle. This is known as stationary phase. Stationary phase is a point in a cell's life when it's not actively growing or dying. And despite the fact that a cell spends most of its life in this phase, it's almost entirely ignored by evolutionary studies. Now, stationary phase can arise for a lot of reasons: lack of nutrients, overpopulation, but my project focuses on oxidative damage, which is when an oxygen atom is missing an electron, creating a reactive oxygen species, or ROS. ROS is bad news for a cell because it tends to damage DNA. And this is where proteins like MFD, or mutation frequency decline, come in, whose goal is to make sure DNA is running at maximum capacity. Now, the function of transcription is essential for a cell's survival. However, damage to DNA can stop transcription dead in its tracks. Under growing conditions, MFD sees this, signals for repair to happen, repair occurs as planned, and the cell is allowed to continue on its way. However, during stationary phase, the mechanism of repair changes. MFD comes in, gives the bat signal for the repair, and the repair is done. And although the DNA is left functional, it's different than it was before. This means a mutation has now arisen despite the fact that the cell actually didn't grow. This presents a huge advantage for bacteria. They don't have to grow to create newer, better versions of themselves. And we believe it contributes heavily to antibiotic resistance. I already have data indicating here that our bacteria, YB955, with the presence of MFD, has a much greater ability to form resistance to the antibiotic rifampicin, which treats that minor thing, tuberculosis. I was like, so ultimately what we're looking at here is that these proteins have a huge effect on how bacteria are changing in our bodies, in our environments, and in the world around us. However, this mechanism of evolution is ignored, but we need to look at it in a way that allows us to target it and study it because it's changing the way we view evolution as a whole. Thank you.
All right, it got quiet, so you must know that the judges have weighed in and we have tallied, which means I know who has won the inaugural Rebel Grad Slam. And if you're good, I'll tell you. First of all, I want to thank again our judges for all of your hard work. I have to say I've seen all but one of these talks, some of them multiple times this week, and every one of our contestants just did better and better and were more and more impressive all week as the week went on. So before I announce any winners, I just want to say thank you and congratulations to all of you. And just to draw it out another moment, we promised an award, a pizza party, in fact, to the college that had the greatest number of participants in our event this year. We're really trying to stir the pot and get the colleges to compete a bit. So this year's pizza lunch will go to the College of Liberal Arts, who had the greatest number of contestants this year. I'm sorry, the provost, I think, wants to offer...
You didn't index by size?
You had to ask. We had this debate. It's on the list for maybe next year. Okay, so now I will announce the winners. You should know that our competitors have been receiving prizes all week that have grown and sort of built up in size and stature. And today, our three finalists are walking away with scholarships. So our third place winner tonight will get a $500 scholarship towards their studies here in their graduate program. Our second place slammer will get a $750 scholarship. And our first place slammer gets $1,000 towards their study here at UNLV. So our third place slammer from the College of Science is Chris Hardy. Please come up. There you go. I'm presenting you with your scholarship.
Thank you very much.
Congratulations. Oops. Show the check. You can just step up here. All right. Our second place award winner in the Rebel Grad Slam from the Department of Anthropology and the College of Liberal Arts is Caitlin D. Benedetto. See, you get a real check. Congratulations. Join us up front. And last but not least, first among our slammers in our inaugural year from the College of Engineering, we have Ms. Erica Murty. Congratulations. Thanks. Join us. Oh no, we're not done yet. We also promised that the college of the winning slammer would take the Rebel Grad Slam Stash Award, and it would be housed in that winner's college for the year to come back next year and move on to the new college. So I proudly present the Rebel Grad Stash to the College of Engineering for 2014-2015.